This is the fifth and final video in the Biology Key Skills revision tutorial series. In this video, we will be looking over the food tests. It is important to note that this is triple or separate science only. If you are doing combined science, you do not need to watch this video. In this video, we will look at the major biological molecules that make up food. We will look at how a calorimeter can be used to measure the energy content of food, and we will look at the chemical tests that can be used to identify a range of biological molecules. For our big question today, 0.5 grams of food is burned in a calorimeter. It causes 10 centimeters cubed of water to increase in temperature by 20 degrees C. We need to calculate the energy content of the food in joules per gram. For your GCSE, you need to know three key biological molecules. These are carbohydrates, proteins, and lipids. Carbohydrates are made up of sugars connected together. You need to know about reducing sugars. These are simple sugars, for example, glucose, and much larger carbohydrate molecules, for example, starch. We find carbohydrates in lots of foodstuff, for example, bread, chips, and rice. Proteins, on the other hand, are made up of amino acids. They are found in all meat products, for example, steak or chicken. They are found in fish, in eggs, and in beans. Finally, we have lipids, or fats. These are made up of fatty acid and glycerol. We find lipids in things like butter or cheese or even in chocolate. For your triple or separate science GCSE, you need to be able to test for each of these biological molecules. When you are testing for carbohydrates, you need to know two separate tests. First of all, we need to know one for testing simple carbohydrates, for example, the sugars. These include glucose, sucrose, fructose, and lactose. To test for this, we add Benedict's reagent, which is blue, into the test sample. We then heat the test sample in a water bath at 75 degrees Celsius. If the sugar is present, the sample will change colour, as it will form a precipitate. The colour that we see is dependent on the concentration of the sugar in the solution. So for example, if we only have a small amount of sugar, then the solution will go green, up to a very strong solution which goes brick red. This test allows us to see how much reducing sugar is present in a solution. For more complex carbohydrates, which are made up of multiple sugars stuck together, for example starch, we need to know a second test. First of all, we add iodine solution, which is a browny orange colour, to the test sample. Iodine solution is iodine dissolved in potassium iodide solution. If starch is present, then the sample will change colour from this browny orange colour to a much darker blue-black colour. If, however, no starch is present, then it will simply stay that brown-orange colour. So again, we get the colour change, we get that positive result if the molecule we are testing for is present. The second test we need to know about is how to test for proteins. This is done using the Biuret test. To start off with, we have our chopped up food in the solution, or our solution which we wish to test. We then add in our potassium hydroxide solution. This makes the test sample alkaline. We then add in some copper sulfate solution, which is bright blue. If the protein is present, then the solution will turn purple. If, however, there is no protein present, then the solution will stay blue. Once again, that positive result, the colour change from blue to purple, indicates that there is protein present. Finally, we also need to be able to test for lipids. To do this, we add the test sample to a boiling tube with the ethanol. We then shake it for a minute until the sample dissolves. We pour this ethanol solution into water, so we mix it with water. If lipids are present, then they will precipitate out of the liquid and they will show up as a milky coloured emulsion. The more lipid there is, the more obvious this milky colour will be. Once again, we are only interested in the positive result from this experiment. If we do not get any milky coloured emulsion, then we simply have no lipids present. It is important to note that an emulsion is when one liquid doesn't dissolve in another and it just forms droplets, a bit like if you shake oil and water. Not only can we test for the biological molecules in food, we can also test to see how much energy it contains. Basically, how much energy is given off when the foodstuff burns. In order to do this, we need to use a food that will burn easily. This is usually something that's dry, for example, pasta, crisps, or bread. First, we need to measure the mass of the dry food, and then we skewer it onto a mounted needle. This will allow us to hold it underneath the boiling tube with the water and the thermometer. Next, we add in our set volume of water to our boiling tube, and we hold it in place with a clamp. This will be used to measure the amount of energy that's transferred or released when the food is burnt. Now we measure the temperature of the water with our thermometer so that we can get a starting temperature. We then set fire to the food using a Bunsen burner before turning the Bunsen off and moving it away from the apparatus. This will make sure that the heat of the Bunsen does not affect the starting temperature of the water. Once the food is alight, we hold it underneath our test tube until it goes out. We then relight the food and hold it under the tube again. We keep doing this until the food simply won't catch fire again. We then measure the temperature of the water a second time so that we can work out the overall change. The whole point in carrying out calorimetry is in order to work out how much energy the food contains. So far, we have got one piece of information, which is the temperature change of the water. We will now look at two equations which will allow us to work out the overall energy that has been released, and therefore work out the energy per gram of the food. Our first equation will allow us to work out the overall energy in the food. Here we have the energy in the food in joules equals the mass of the water in grams times by the temperature change in degrees Celsius times by 4.2. The mass of the water is calculated using the volume, where 1 centimetres cubed of water is the same as 1 gram of water. The temperature change in degrees Celsius is worked out by taking the initial temperature of the water and taking that away from the final temperature of the water. Finally, we have this value of 4.2. This is the specific heat capacity of water. This is the amount of energy needed to change the temperature of 1 kilo of water by 1 degrees C. Different substances have different specific heat capacities. For water, this is 4.2. Once we have a value for the energy in food in joules, we can then use our second equation. Our second equation allows us to work out the energy per gram in the food. To do this, we take the value we calculated in Formula 1, which was the energy in food, and we divide it by the mass of the food that we recorded right at the beginning of the method for the practical. By converting from the energy in food in joules to the energy per gram in food of joules per gram, this allows us to directly compare the energy values of different foods. It is also important to note that quite a bit of the energy in the food is actually transferred to the environment and not to the water. We can minimise this by insulating the boiling tube, for example, with foil. So, what have we looked at in this tutorial video? Well, we described the major biological molecules that make up food. We looked at how a calorimeter can be used to measure the energy content of food. And finally, we looked at how chemical tests could be used to identify a range of biological molecules. Our big question today is that 0.5 grams of food is burned in a calorimeter. It causes 10 centimetres cubed of water to increase in temperature by 20 degrees C. We need to calculate the energy content of the food in joules per gram. I want you to have a go at that question now using both formulas that we have just looked at. So, for this question, we need to use both equations. The first equation we're going to use is the energy in food equation. So energy in food in joules is equal to the mass of water in grams times the temperature change of water in degrees Celsius times by the specific heat capacity of water, which is 4.2. From the question, we were given 10 centimetres cubed of water. As 1 centimetres cubed of water is the same as 1 gram of water, we have 10 grams of water. Our temperature change is 20 and our specific heat capacity is 4.2. So we're going to do 10 times 20 times 4.2, giving us 840 joules. We now need to use equation 2, which was energy per gram of food in joules per gram is equal to energy in food in joules over the mass of the food in grams. We've got the energy of the food, which is 840 joules, and from the question we were given the mass being 0.5. So we do 840 divided by 0.5, giving us a final answer of 1,680 joules per gram. That concludes not only this tutorial video, which was tutorial video 5, but also the entire Biology Key Skills tutorial series.